Good morning, children. How are you all? So now it's the second class of nutrition. The first class we have learned what is nutrition, what are the different kinds of nutrients, and what are they. And also we have learned about the autotrophic nutrition, and also we have learned what is the photosynthesis and what is the overall equation of photosynthesis. So as we all know that this is the overall equation of the photosynthesis. You can see 6CO2 plus 12H2O gives rise to 6H2O6 plus 6O2 plus 6H2O. And in the last class we have done this experiment, that is how to test the presence of starch in a leaf. Already we have discussed that leaf is the food factory of the plant. So definitely the leaf will have the starch in it. So we have seen a simple experiment to make sure that the leaf has starch in it. So if it has leaf, if it has starch in it, you will know it. If it doesn't have a starch, you will know it. So one experiment we have already finished. So now we will try to see the experiment of carbon dioxide, which is also a very common question in the examinations. So this carbon dioxide, whether it is essential for the process of photosynthesis or not. So we have to make sure that carbon dioxide is essential for photosynthesis because that is written in the equation. So let's go ahead. So carbon dioxide is present in the air. So the carbon dioxide is present in the air, and the leaves have to take this carbon dioxide through stomata which are present on the leaf. So now we will base our experiment completely on the same. Now to prove the carbon dioxide is essential for photosynthesis or not, what we are going to do is we are going to take a potted plant. We are going to take a potted plant, and when you take the potted plant, make sure that it has long and narrow leaves like this, like you have seen the mango leaves, right? So these are the long and narrow leaves. So you have to select such a plant, and this potted plant also should be kept in dark for at least a week so that the leaves will get de-starched. What do you mean de-starched? The leaves will not have any starch. How do you know that they don't have any starch in it? It's very simple. Take the leaf, okay, keep it in the test tube, fill it with the methyl spirit, and then keep it in the beaker which contains the water. Take a Bunsen burner, heat it, boil it. It will become pale, and then you keep it in the petri dish and treat it with the iodine solution. So when you treat it with the iodine solution, if it turns into deep blue color, then it has starch in it. If it doesn't turn into deep blue color, it doesn't have any starch in it. So it's very easy to do this experiment. That is what we have learned in the starch experiment. So now here also I am taking a potted plant here. Which is kept in dark for at least a week of time. So potted plant. Second one is that should have the de-starched leaves, and second one is they should have the long and narrow leaves. Okay, so this is a basic need. Okay, so if you take a starched plant, if you take any other leaf like that, what will happen is you will never will be able to do this experiment. So only we have to take the de-starched plant, right? After that, we are going to take a wide-mouthed bottle. What we are going to take? We are going to take a wide-mouthed bottle where you can place this leaf. The leaf should be able to go into the bottle. So you have to take a wide-mouthed bottle. What we have to take? White You have to take a wide mouthed bottle. Then what we have to take is, we have to take a cork. We have to take a cork. The cork should be in such a way that it fits into the bottle like this. Okay, we have to take a cork like this, and moreover, the cork which you are taking, you have to split the cork into 2 equal halves. You take a knife, cut that cork into 2 equal halves so that you can insert the leaf. Okay, so this is what we have done. So the next thing we have, which we have taken, is the cork, which is split. Now we also will take the solution which is called as KOH solution, which we called as the potassium hydroxide solution. So we are taking the potassium hydroxide solution here, and after taking the potassium hydroxide solution, what we will do, we will make sure that we will put this potassium hydroxide solution in the bottle. Okay, so now how to arrange this apparatus? How to arrange this apparatus we will see now. Okay, so to prove the carbon dioxide is essential for photosynthesis process, what are the things we have taken? What is the apparatus we have taken? So one, we have taken the potted plant here, and the potted plant should have a de-starched leaf, and basically select the plant which has the long and narrow leaves. If you take the broad leaves and all, it might not be useful for our experiment. So better you take long and narrow leaves, it's mandatory. And then we have taken a wide mouthed bottle like this, okay, a wide mouthed bottle, so that we can insert the leaf like this, okay. And then we have taken a cork, and we have split that cork into 2 equal halves so that we can insert the leaf through the cork and close the mouth of the bottle with the cork. So now we have also taken a stand so that we can make the bottle stand straight, and also we have taken some kind of support here. So this is how you have to arrange the apparatus. Okay, potted plant, wide mouth bottle, a split cork, and then something to support the bottle. And moreover, what we have taken is the potassium hydroxide solution. What is the solution we have taken here? We have taken the potassium hydroxide solution, so that right now we finished this complete setup. So now we have to keep the same apparatus out in the sunlight, because for all this week this particular plant did not receive any sunlight, so the leaves are completely de-starched. It doesn't have any kind of starch in them. So now we are putting this apparatus in the sunlight. We'll wait till evening, and then what we will do is we'll bring the apparatus. And now we doesn't require the complete apparatus, right? So what we want only is the leaf. So detach the leaf from the plant. So once you detach the leaf, then we have to go ahead with the experiment of starch. So the same way, take this leaf, put in the methylated spirit in a test tube, keep it in the water, take a Bunsen burner and take some water in the beaker and heat it, and after that this particular leaf will lose its chlorophyll, and then it will become pale in color. And then you have to take this in a small petri dish. Okay, let me draw it here. We'll take the leaf in a petri dish, and when you do this petri dish experiment, what will happen is only the part which is exposed outside the bottle will show the presence of starch. It will show the presence of starch, but whereas the part of the leaf which is inserted in the bottle, this particular part will not show any color. What is the reason behind this? The reason is potassium hydroxide solution which you have taken in the in this beaker. What will it do is it will absorb the carbon dioxide which is present in the bottle. So it is making the complete bottle carbon dioxide free. So once the bottle becomes carbon dioxide free, the leaf, this particular part which is in the bottle, will not get the carbon dioxide. So as it is not getting the carbon dioxide, so this particular part is not able to prepare the food material. If it had got the carbon dioxide, then it would have shown the blue color, but it's not showing any kind of color here. The reason is it did not get the carbon dioxide, because whatever the carbon dioxide which is in the bottle, in the white mouthed bottle, is completely absorbed by the potassium hydroxide solution or the pellets we have taken in the bottle. Now second thing, you might get a doubt that maybe some of the air might have escaped into the bottle because we have taken a split cork. So for that also what we will do is we'll put some Vaseline on the corners of the bottle where the split cork is inserted, and even at the cork where it is split, so that will make sure that the air from the atmosphere will not enter into the bottle. And moreover, the second precaution what we have to take is we have to make sure that the leaf will not touch the potassium hydroxide solution. So if you take all these measures, then when you go ahead with this experiment, in the starch experiment, you can see that only this particular part which is outside the bottle will turn into deep blue color, but whereas the leaf which is in the beaker or in the glass bottle, white glass bottle, will not turn into deep blue color because it did not have the carbon dioxide. So it did not come in contact with the carbon dioxide. Now what we are able to prove here, so plants require carbon dioxide at any cost. It is essential for the photosynthesis process. If there is no carbon dioxide, plants cannot prepare the food material, though it is getting water, sunlight, chlorophyll, everything, but still if carbon dioxide is not there, plants will not be able to prepare the food material. So this is the experiment which we have learned in the photosynthesis equation. Okay, now, okay children, that's all for today. See you tomorrow with oxygen experiment. Till then, goodbye.